Hi, I'm Trung Tran. I'm a PhD candidate in computer science at Duke University, advised by Alex Hardemink. In our lab, we're trying to understand the relationship between the chromatin state and gene expression. The chromatin state is a complex array of DNA binding factors, proteins, polymerases, and many other factors that interact with each other, bind and unbind to the DNA, and basically its state will determine what any type of DNA dependent transaction. So in this poster, I will be trying to understand the chromatin state in relation to gene expression using a vision transformer model. So to start, we use an assay called MNase, and in that we use an enzyme called MNase, which is an endoexonuclease that will cut unbound DNA and digest up to any type of DNA binding proteins. And the result is an image like this, where on the x-axis is the genomic position and the y-axis is the fragment length. So larger fragments, like these nucleosomes here, will be represented in this dot plot as larger fragments between 140 and 160 base pairs, and smaller fragments, such as transcription factors, will be placed on the y-axis as smaller fragments below 80 or 100 base pairs. So with these type of images, we have these type of two-dimensional plots that we can create images out of and feed them into a type of model called a vision transformer. So vision transformers allow you to partition and linearize those images and then feed them into transformer encoder blocks. These transformer encoder blocks will summarize these images as using a self-attention scheme, and those attention schemes can then be used for inference. So once prediction is made, where the prediction is transcript level, we can use those attention weights to draw inferences that will inform us about which parts of the input chromatin images are most relevant to our prediction. The dataset we'll be using is this cadmium perturbation assay, in which we have 6 time points. After the first time point, cadmium is introduced, which induces a transcriptional response to handle the stress caused by cadmium, the heavy metal. In each time point, we collect the chromatin state and the gene expression using MNase-seq and RNA-seq. And using these pairs, we can use the chromatin state to try to predict the gene expression level. And for the first time, 5 time points, we'll be using that as a training set. We will omit any type of time label and just have a shuffle of various genes and the chromatin state at each of these time points, and then try to predict the time point 120. The end result is that we have a very highly predicted, well-predicted time point of 120, and we can then determine what parts of the chromatin are most relevant in the prediction. So if we were to look at RPS7A, one of the genes that are downregulated in this cadmium response, it's a ribosomal subunit protein, and most types of translational type pathways are downregulated under these stress conditions. We can look at the attention weights here, where our input MNase image shows in the promoter the small fragments enriched, and a +1 and +2 nucleosomes are fairly well positioned and kind of upstream of the transcription start site. In our attention weight mask, we see the +2 attention is what's most relevant in the prediction for RPS7A. We can then take these attentions and genome-wide for time point 120, we can cluster them to see if there's commonalities between different attentions that are being used to drive prediction. And here we have the attentions in each of the clusters, and to the left we have the time course of the transcript level. So you can see each of the 6 time points, although the attentions are what are being clustered are just for time point 120. And then for the third column, we have the chromatin state. That is the MNase images for each of the clusters composited into one image. And what you can see here is that these the earlier clusters in which the transcript level is low for time point 120, the relevant attentions are actually the -1 and +1, or possibly the +2 and +3 nucleosomes. While once you start getting into higher transcript clusters, the more relevant features are within the promoter of the gene. So the smaller fragments, 100 to 200 base pairs, or even 300 base pairs upstream of the transcription start site, are what are relevant in the prediction. And this allows us to kind of determine where these small fragments are being enriched. We can perform sequence analysis or motif analysis and maybe determine if there are relevant annotations or adjacent genes or other types of genomic features that might be relevant in informing the gene prediction. So further analysis is definitely going to be required to understand these attentions with more detail. And we can extend this model to other types of data, such as including sequence, other genomic features like histone marks, or whether or not these genes have divergent or tandem genes nearby. So there are a lot of extensions we can take with this model, but this is kind of a baseline to determine if this might be an interesting direction to study the chromatin and possibly determine if there's any biological relevance to these key features that might explain gene transcription. Thank you.